Hello, everyone, and welcome to today's live broadcast. A well-implemented LIMS equals good data integrity. I'm Alexis Cross of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Sartorius. For more information about our sponsor, please visit their website at sartorius.com. Now, let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, please report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenter, Siri Segalstad, the lead consultant at Segalstad Consulting. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Siri, you may now begin your presentation. Welcome.
Oh, thank you very much for the introduction, and welcome to this presentation on how LIMS can help you with data integrity. I believe that most LIMS on the market have the technical functions to help. The big question is if it has been implemented correctly, and that depends on how the users have implemented their LIMS. During this presentation, I will first talk about what data integrity is and how it's defined, and then I will talk about how LIMS can help. My name is Siri Segalstad. I live in Norway. I have a degree in chemical engineering and analytical chemistry. I work with LIMS and quality assurance since 1987. I worked as a consultant since 1995 with LIMS, quality assurance, validation of IT systems, data integrity, and a lot of other things. I've also participated in the EU Leonardo da Vinci project to create a master's degree in IT validation. And I taught my classes at the University of Ghent, Belgium. In the Leonardo da Vinci project, we were supposed to create the classes based on existing books, but there were none that covered what I needed, so I wrote that after that 3-year project was finished. Data integrity would have been easier if there was only 1 or 2 standards, but there are many, and they're also frequently upgraded. This is an overview, which I've included with permission from Chris Burgess and Bob McDowell. As we can see, the pharmaceutical regulators in both the US and the EU have created their share of requirements. Additionally, there's text more or less hidden in all of the GMP, GLP, GCP, medical devices, etc. regarding how to document what is done. The probably best document to start reading is the World Health Organization, WHO, Good Data and Record Management. The newest version is from 2016. Other requirements documents are from Pharmaceutical Inspectors Convention, PICS, and there are also a lot of guidance documents, for example, from GAMP, PDA, European Compliance Academy, and APIC. ISO 17025 and other standards also have focus on data integrity, and not the least, the pharmaceutical regulators have data integrity high on their inspection plans these days. I've added a few pictures from my beautiful country, and I hope you will enjoy them. First, in this presentation, I will talk about the terminology related to data integrity, records management, and data governance. To get into compliance is a long and winding road. We can start by looking at the definition of data integrity. Here's from the World Health Organization, WHO. They state that data integrity is the degree to which data are complete, consistent, accurate, trustworthy, and reliable, and that the characteristics of the data are maintained throughout the data lifecycle. This includes ALCOA-plus CCEA. Assuring data integrity requires appropriate quality and risk management systems, including adherence to scientific principles and good documentation practice. Well, we see that this includes some cryptic letters, ALCOA-plus CCEA. So what do those mean? ALCOA-plus is the acronym that covers the data integrity and what that is all about. It's frequently written as ALCOA-plus with a plus sign, and the plus includes the CCEA. So what are these letters all about? ALCOA is A for attributable, legible, contemporaneous, original, accurate, plus the CCEA, which is additional emphasis on these attributes, all of these attributes, being complete, consistent, enduring, and available. Well, they're still just words. I will explain them, or actually, I will have some help from WHO Annex 5 and 21 CFR 211 to explain them. The A in ALCOA is for attributable. And the WHO states that information is captured in the record so that it's uniquely identified as executed by the originator of the data. That could be a person or a computer system. 21 CFR 211, the GMP, the US GMP, states that attributable means identification of the individual who performed an activity. And here we're actually digging into the audit trail, which should be well known to anybody who's worked with electronic records in the pharmaceutical industry the past 25 years. Mind you, there's no requirement for using a computer system, so even paper records need to include this correct information. The L in ALCOA is for legible. And here we have a fantastic, enormous big sentence from the World Health Organization. Data are reliable, understandable, and allow a clear picture of the sequencing of steps or events in the record so that all GXP activities conducted can be fully reconstructed by the people reviewing these records at any point during the record retention period set by the applicable GXP. The 211 has a more pragmatic approach. Can you read the electronic data with any associated metadata or all the written entries on paper? Legible should also extend to any original data that has been changed or modified by an authorized individual so that the original entry is not obscured. Now we get to the C. It's for contemporaneous, which means at the same time. And that is exactly how WHO explains it. Contemporaneous data are data recorded at the time they are generated or observed. And GMP states that documented on paper or electronically at the time of an activity. Both of them agree. This means that it shall be documented while you're working on it and not later. Now we are at the O, which stands for original. Original data includes the first or source capture of data or information and all subsequent data required to fully reconstruct the conduct of the GXP activities. And there's more in the details in the text. The GMP states that it's a written observation or printout or a certified copy thereof or an electronic record including all metadata of an activity. Now, this means really that you cannot print out the results, fill in the data in another table, and throw away the printout. Furthermore, you will have to have documentation for all the surrounding data that was needed to get this result. That means that you have to be able to recreate it completely. I'll explain more later which data we're talking about. Finally, the second A in ALCOA, which is for accurate. And WHO states that the term accurate means that data are correct, truthful, complete, valid, and reliable, whereas the GMP states that no errors in the original observation or observations, no editing without documented amendments, audit trail entries by authorized personnel. Now the plus, which covers the CCEA. These are implicit in the requirements in ALCOA. The record shall be complete, consistent, enduring, and available. Complete we will look a bit more into later. Consistent is that things are done and documented the same way, and enduring, the data will last, that is, stored in a secure way, and they must be available. This is all quite complicated, especially if we use a paper system. We will need we will need logs for which reagents and standards we used and how they were made. We will also need logs for instrument, maintenance, calibration, use, and of course, which of the same type of instrument we used. The conclusion is that we do need a LIMS. So what is a LIMS and how can it help us? First of all, a LIMS is a laboratory information management system, and it can take care of all the data in the lab if we let it do that. The lab needs a LIMS in order to record everything that's prescribed in ALCOA-plus. The LIMS must be implemented so that all metadata is available inside the system, no shortcuts. And metadata, I will explain. There should be electronic transfer of data between LIMS and instruments, and I will talk later about where the original data shall be stored. First, I would like to talk about static and dynamic data in LIMS. They are frequently called something else in the different LIMS systems, but I think you will understand which is which, even if it's a different name that you're using. The static data is that the lab tells LIMS how the lab operates. That includes definitions of sample types, studies, analytical methods, calculations in the methods, instruments, reagents, and standards, and sample specifications, users and their access, to mention a few of the static data. We will have to populate the database with our data and our way of working. When we start using the LIMS live, we create dynamic data for this sample, this instrument, these reagents, etc. And LIMS will tell us how and which data we shall collect based on the static data that we've already created. The static data are implemented in LIMS, which means added to the LIMS before we can use it for our samples. When that has been done, we can add this sample with batch number and so on, and the dynamic data are created for this sample. If we try to explain this from a paper-based lab, static data would be the analytical method descriptions and the specifications for the sample type, the instrument SOPs, and the reagent SOPs, to mention a few. Dynamic data is the result for this specific sample or batch, the logbook for the instrument, etc. Of course, there are many more dynamic data that will follow this sample to make it the way the ALCOA-plus describes. Before LIMS can be used, the tables must be populated with our static data for workflows, sample types, studies, instruments, analytical methods, specifications and other limits, and the defined users with their specific access. And all of these must be implemented with care to fulfill the data integrity requirements. Let's look at an analytical method or two and see how we can implement them in order to be in compliance. Because the ALCOA-plus is a lot more than documenting the measurement itself. We must, for example, be able to prove that we have and had control over the instrument and its data, and we must have all the metadata in order to be able to recreate the data later. Metadata? Well, this may be a new expression, but here are some examples. First, let's look at reagents or standards or chemicals. The original bottle or jar that's purchased from a supplier. We create this in LIMS so that we have the following information: name, purity, concentration, manufacturer, manufacturer's batch number, internally given identification, however that is done. And when all this is entered, we will let LIMS create a label which has the relevant information, like this label here. Note the barcode on the label. This is useful when we shall use the chemical later. Now, we didn't need 35% hydrochloric acid, or, as the sample here, some dry chemical. We need a thinner solution, or even just a solution. And our analytical method, it states, weigh approximately 2 grams accurately and dissolve this to 100 milliliters. We will include this preparation in LIMS. First, we scan the original on the last page, so LIMS knows exactly which one we have used. Then we weigh these 2 grams, and I always thought it was funny to have approximately and accurately in the same sentence. But anyway, we will weigh these 2 grams. The balance is connected to LIMS, so that we will send the weight that when we're happy with our 2 grams, and LIMS knows that the volume is always 100 milliliters and will then automatically calculate the concentration. LIMS will also give this new reagent an ID and print out the label like the one on the last slide. Of course, LIMS will have to be told first how to handle this as static data. When we shall use the reagent, we can scan the label, and LIMS knows exactly which reagent we have used and the concentration. This is often the input to an analytical method and may be part of the calculations in this method. We weigh things all the time in the lab. Weight in itself is usually not a method, but it's a part of a lot of methods. Weigh the sample and do, well, whatever. This means that we need a LIMS that can handle connecting at least 2 instruments to the same analytical methods. Some years ago, not all the LIMSes were able to do that. I think they are now, but add that to your user requirements if you're planning to buy a new LIMS. In fact, all instruments need to be defined in LIMS, and the static data includes the instrument information, type, model, serial number, etc., maintenance information, frequency and what to be done. This is a SOP requirement. When was it last done? When is the next time, or a due date? That's the instrument logbook. And can the instrument be used after the due date? It will be flagged if it has been used, if it is allowed to use it. Calibration information is the same as for maintenance. That was on the last slide. The next question is, do we need some sort of system suitability test before use? In that case, we need to create a method for that. How is this instrument connected to LIMS? 1 way? 2 ways? Connection to LIMS depends on how complex the instrument is. Simple instruments may only be able to send information to LIMS, but not receive from LIMS. So the 1 way is always from the instrument to LIMS with results. More complex instruments and all computerized instruments can have a 2-way communication with LIMS. Here, LIMS tells the instrument about method, sequence, sample identification, standards, control samples, etc., and receives the results for each sample when that has been analyzed. Calculations may be done by the instrument itself or its computer system, or we can let LIMS do that. For example, the HPLC with its chromatography data system will do all the calculations and will send the final result back to LIMS and keep the raw data in the chromatography data system. Whereas a balance may send the measurement to LIMS, and LIMS will do the calculation in the analytical method based on weight and other factors. I mentioned that data may be stored in LIMS or in the instrument. How do we decide where to store it? In general, this is the first point of entry to a software system that can store and handle the data. So the rule of the thumb is that LIMS must store all data that the instrument cannot store. For example, balances and simple instruments without computers. LIMS may also have file servers, for example, for pictures from microscopes. A computerized instrument is generally more suited to store a large amount of raw data and to do the calculations for these complex instruments. And examples would be chromatography data system, atomic absorption, ICP, MS, or those hyphen systems, GC, MS, etc. However, we must make sure that the computerized instruments and downloading their data to a server so that the data is backed up. No solitary instrument shall exist in the lab. Network is alpha and omega if we want to have our data integrity. So hook them up. Now we have several balances in the lab, and each of these need to be defined in LIMS, but they also need some dynamic data. When was it maintained last? How long before the next maintenance? Same for calibration. We have to set up the static data, so it is not possible to use the instrument unless it has been maintained and calibrated. Or we can set it up so that we can use it, but then the result will be flagged as obtained by an instrument that has not been calibrated or maintained. This is based on the dynamic data with input each time something happens to the instrument. Now we can add the instrument, this specific instrument, to the weight data we receive when weighing the sample. The previous balanced logbook is now in LIMS and is available at our fingertips, ALCOA-plus. And so it goes for all possible metadata. Implement everything, don't leave anything out that has relevance to data integrity. So let's talk about implementation of analytical methods in LIMS. pH methods are simple. You measure the pH, make a note, and that's it, right? No, wrong. With data integrity in mind, you will need to have a lot more information. You need to know which instrument was used, calibration and maintenance, was that okay? Which electrode was used, status, age, checked, etc. Was it okay? Which standards were used, and if they were diluted, how were they diluted? Did we use control samples? Well, then we need to make a note of those as well. And then we can do the sample measurements. So pH methods are not simple if you want to talk about data integrity. Here's another, Carl Fisher, where we titrate to find water content of the sample. When we enter this method as static data, that is, implement this in LIMS, there are several considerations for how to do this. We need a sample weight measured in grams, which we get from a connected balance. When we set this method up as static data, we can either just state that we need a balance and have the operator input the name of the balance used, or we can say that we always use this specific balance, which the operator can or cannot overrule if that is necessary, and the can or cannot is something you set up. We enter the type of standard that we use so the operator can scan the label on the standard. That will give information about the concentration of the standard based on the ID of the standard. We scan the barcode. We enter that we will use a Carl Fisher instrument. If we had only one, we can link that one directly. But if we have more, do the same way as with the balance. The instrument will give the titration volume when the method is used for a sample. Finally, we set up the calculation formula so that LIMS can calculate the results based on concentration, weight, volume, and a conversion to percent. This is what it will look like in LIMS when we use the Carl Fisher method. Of course, each LIMS will have a different layout, but the basic information is the same. We have logged the sample 2436A in this case. LIMS knows that this sample shall have its water content determined by the method called water content by Carl Fisher. Weight is entered electronically from the balance, and LIMS then knows which balance and its calibration information. We scan the barcode on the standards bottle, and LIMS knows the concentration, expiration time, and other information. We do the titration, and the volume is entered by the Carl Fisher instrument, and LIMS knows all about that instrument as well. LIMS will then calculate the water content for the sample based on the calculation formula we have entered. There is no need to use calculator or Excel, LIMS does it. And as we have validated the method and its calculations, we know that the calculation is correct. All the metadata is available, and we have the following ALCOA: attributable, legible, contemporaneous, original, accurate. And the plus factors follow also as we back data up from LIMS so that it is complete, consistent, and durable, and available. Now a little on the dynamic data. As we have entered the static data for all our sample types, instruments, reagents, methods, specifications, etc., we can use LIMS for the daily use. We have also connected the dots so that LIMS knows that this sample type needs these analytical methods and have this defined specifications. As the analytical methods are also connected to our reagents and our instruments, we have a good overview of our data and where they come from. We receive a sample that shall be analyzed. This has already been registered in LIMS and has received a label with batch number and other information. So we scan the label, and LIMS now knows what we shall do with the sample. LIMS will send the message to the instrument to be used, for example, the HPLC chromatography data system. This may also include run information in the method to be used, and the sequence of the standards, control samples, blanks, and samples. LIMS will receive the analytical data from the instruments. From the CDS, chromatography data system, there will be results that have already been calculated based on the method standards, etc. For other methods, like Carl-Fisher, LIMS will calculate the result. LIMS will then compare the result with the specifications for the sample and will flag if these are okay or out of spec. We have also set up LIMS to approve results automatically if nothing has been flagged. Flags can be set for maintenance, calibration, expiration dates, problems with system suitability testing, out of range for instruments, out of spec for results, to mention a few. A GXP sample should always have a final approval by a qualified person like someone in QA, and LIMS does have that technical function for electronic signature. I have a few general thoughts regarding methods. When I implemented our new LIMS in 1989 in a pharmaceutical company in Oslo, we had to set the number of digits globally for the whole LIMS system. We chose 5 as that was the maximum that our balances could do. However, it did not make any sense to enter the number of dead mice with 5 digits. 1 digit perhaps, like 1.5 dead mice, 1 dead and 1 half dead, but that was about it. In today's LIMSes, we can enter the number of digits for each measurement. Balances have a fixed number of digits each, so that number is what we use when entering a weight. But LIMS also makes a lot of calculations, and we need to have the correct scientifically determined number of digits based on method validation. Don't forget that it shall be scientifically sound. Otherwise, you're showing an accuracy that you don't really have. Visual inspection methods are manual and not determined by instruments. In this example, we have a spec stating sample shall be white to off-white fine-grained powder. How do we implement this in LIMS? Well, we can say pass-fail. Well, pass is okay, but what failed? Let's try pass-fail question for each requirement. No, we shall not. Pass-fail is an assessment. It is not a result. We shall implement this as observations for each of the requirements. After all, it's a visual inspection method. We do not observe pass or fail. We observe if the sample is white, off-white, or another color. We observe if this is a fine-grained powder, however that is defined. So what we do is to create lists of possible answers of color, white, off-white, yellow, green, or whatever we need. The user shall choose one of these when entering the answer. In that way, we also avoid typos. We can also create a list of grain, fine grain, lumps, coarse grains, etc. And finally, we will let LIMS make the assessment based on the specification. Sample shall be white to off-white fine-grained powder. If we later have to change the spec and run the sample against the new spec, we can see if it is still good. You cannot do that with a pass-fail result. Another thing is that we may want to run trends on the sample, and a trend with pass, pass, pass is not very useful. A trend with white, white, off-white, white gives information. Electronic signatures. Those are used in systems instead of handwritten signatures on paper. That means that the status is signed. It's used in LIMS where requirements, for example, the GXP, say that the item shall be signed. And please do not add more electronic signatures than necessary. The audit trail will take care of other information, like who did what. We don't need a signature saying that I did this. The audit trail will do that. And the explanation for an audit trail, for those who are not familiar with it, each user is defined in the system with his or her user ID and access to functions and data. The user has to log in with his or her own credentials before the system gives access to the person. The audit trail is automatically added to every entry in LIMS and to every change in existing data. The audit trail includes the user ID or full name, date, and time with hour, minutes, and sometimes also seconds. If the user makes a change to existing data, the system shall ask why, and the user must give a reason. These reasons should also be on a list, otherwise you will get reasons like hungover, Monday morning, and Friday evening, which is not very scientific. And believe me, we saw those when we left that open to the user. So create a list. There are specific requirements for functionality in electronic systems for e-signatures, stated in 21 CFR Part 11 and the EU equivalent GMP Annex 11. And the LIMSs will handle that. When you print it out, for example, in a certificate of analysis, use a normal font and state that this was electronically signed. Do not copy a handwritten signature. Don't scan it and copy it. That looks fake. And also, the WHO states that a hybrid approach may be used to sign electronic records when the system lacks features for electronic signatures. However, the use of hybrid systems is discouraged, and use of stored digital images of a person's handwritten signature to sign a document is generally not acceptable. A hybrid system is may be used to sign electronic records when the system lacks features for electronic signatures. A hybrid system is a combination of electronic data and paper data. It's strongly advised not to use those hybrid systems. In fact, the FDA still allows them, but that permission I don't think will last very long. So if you have a LIMS, implement it so that that can take care of everything you need, including electronic signatures. The functionality is there, but you need to utilize it. We have already discussed how to implement various items in LIMS, and LIMS can be perfectly well adapted for data integrity, but it can also be totally useless for the purpose unless you implement it in a good way. I think Lewis Carroll wrote this in a good way when he wrote Alice in Wonderland in 1865, more than 150 years ago. It's old, well-known, and still valid. Which way ought I go from here? Asked Alice. That depends a good deal on where you want to go, said the cat. Oh, I don't much care where, said Alice. Then it doesn't matter which way you go, said the cat. I think this is also very true for a LIMS implementation, and not the least for selecting a LIMS. If you don't state what your needs are, then you will not get what you want. Most of the large LIMSs have been available for a long time and will be okay for your purposes. They are not all that different, but you need to make it work the way it should. Thinking of data integrity from the very beginning of the implementation phase is important. First, the selection of LIMS is based on the written user requirement specification, also called a URS. You will choose the LIMS based on this. However, as each LIMS has so many different ways of solving your requirements, the supplier and you will generally create a functional specification to fit their system with your requirements. There will be several ways of solving them, and the supplier has good knowledge of how their system works. In your URS, you have also written something about your workflow, and this shall of course be implemented as well. Just a little bit of warning based on my own experience. Back in the 80s, we had an R&D lab where people worked on one of 12 different projects. This was the late 80s, before we had any SOPs to speak of. We realized that there were 12 different ways of writing weight percent in the R&D lab. You can see that on the slide. As humans, we easily see that this is the same, but a computer system does not. We decided to use one of them, and people from the 11 other projects were furious about this stupid LIMS that could not handle their way of writing the unit. It was not that LIMS was stupid and couldn't do it, but in order to have some sort of standardization for comparison, reports, etc., we had to choose one of them. One way to make the work better is to standardize. I barely touched on LIMS validation. The topic is actually a long class in itself, and we do not have time to get into details. Validation of LIMS is well understood now. The book I wrote has a lot on validation, and there are plenty of other publications and books as well. I would also like to mention that GAMP5 is an excellent source for IT systems validation. GAMP also has other guidelines for other IT-related pharmaceutical topics. The LIMS itself needs at least these documents. validation plan, risk assessment, user requirement specification, functional specification, implementation plan, DQ, IQ, OQ, PQ, validation report, SOPs. Additionally, each instrument needs to be qualified with roughly the same documents as those listed here. And your analytical methods shall be validated to see how they shall be used, so you can find out about accuracy, quantification limits, and detection limits. Validation is expensive and it is a lot of work. However, not to validate is a lot more expensive. That may mean that you will not be able to get your products on the market or have to withdraw what you have already sent to the market. Or, even worse, that you have to stop production until the FDA is happy with your validation efforts. That has happened. And that takes time, is very costly, a lot more costly than validation before you start using whatever you're using. I took part in the LIMS conferences many years ago. What we heard was that more than 50% of the projects failed. LIMS was not good for them. But actually, that was not the root cause. I've found that there are two reasons why LIMS projects fail. One, there's no user requirement specification. Remember Alice in Wonderland? Two, the management was not involved in this new toy for the lab, which I've heard it being expressed as. That means that when things need changed, like, for example, which system do we use for the unit, or how do we change our workflows and responsibilities, they didn't want to get involved. They may not even tell people that it was mandatory to use this new system. The result was a lot was left in limbo and the project failed. Today, it's a lot better. For once, the technology is better and people are used to using computers for everything. Another reason is that suppliers are involved in the implementation phase, for example, by writing the functional specification together with the users. Our supplier in the 80s rolled out the tape on the deck machine and told us to start implementing, and we did not have a clue about how to do that. It is still possible for a LIMS implementation project to fail, but it is not if we have good people and a good organization around it. We need to have definition of tasks, define the borderlines between LIMS and other systems, and have some sort of culture change management. Well, the same can be said for data integrity. It's expensive. I know two consultants who worked full-time for 6 months on getting the data integrity in a company in a lab actually, in a good condition. That sure is a lot more expensive compared to doing it right from the beginning. There are several tough challenges though, because there are requirements in good records and data management. As far as I can see, there are 4 main challenges. 1, management needs to be heavily involved, and we all know how busy they are. Personnel training and adherence to requirements, well, it might be easier to take a shortcut. Documentations are both electronic and on paper, and how do we manage both in a consistent way? Outsourcing of work and services. The same rules for data integrity applies as for our own work. And how can we ensure that that is good enough? Well, more details follow. Requirements for data integrity is that management shall be involved, and that means a lot more work for them than they had before. They are responsible for good data management. They have to create realistic and achievable expectations, monitor processes. They need allocation of necessary resources for this good documentation practice, both internally and for the outsourced services and suppliers. The adoption of the quality culture, which means it is okay to do errors and to report errors, but no retributions. In many countries or cultures, there is a blame culture. Who can we blame for what goes wrong? In any quality-driven environment, what goes wrong is not who can we blame, but what can we learn from it so we can do it better next time. This is also called CAPA, Corrective Actions, Preventive Actions, and stated in a lot of standards. As the main goal here is not to blame anyone, that will be a very big challenge in many organizations and countries. In some cultures, you cannot make mistakes because it makes you lose face. For someone to admit that they made a mistake before anyone found out may be very hard. There's a lot of active items for the management to do in order to be part of the data integrity. They need to audit and review on a regular basis, make sure that things are always followed up, facilitate the large amount of resources needed for data integrity issues. That includes quality manuals, SOPs, risk and risk management, and not the least, the daily overall management. This will probably mean that more management is needed in the organization. The employees also have a few new challenges. They need 100% adherence to good documentation practice for both paper and electronic records. Most of them are already used to having to follow SOPs for the smallest details, and as such, data integrity is not really news. But I do see that researchers who are used to doing things and scarcely documenting this, especially if work fails so they have to repeat it, they may have a problem adjusting to this new level of documentation. Again, it shall be possible to make errors without retributions. As we are, at least hopefully, used to from the current quality systems, we do know that there is a need for independence in tasks. Time versus quality, for example. If we can't finish the work with adequate quality on Friday evening, we'll just have to wait until we're really done. Doing versus controlling. Those who do the work shall, of course, not control themselves. Computer system access, daily use versus changes in system setup. What do I mean by this? You may have an application manager with full access to everything in LIMS, but he or she is also an analyst with reduced access. That person needs to log out as an application manager and log in again as an analyst before entering data from the lab. The person wears two hats and must be aware of that. I've already said that hybrid systems are not good. Avoid paper if possible. It's hard to keep paper records and electronic records consistent throughout the life cycle. Which record is the original? How do you make sure that the new version of the certificate of analysis is handled correctly? And what about the old version that we already sent out to somebody? Record keeping methodologies and system designed to facilitate ALCOA-plus. That means a zillion SOPs if we have a hybrid system, and a bit fewer if we do this electronically only. And remember, ALCOA-plus applies to both paper and electronic records. The fourth problem is outsourcing. You need to audit the provider before the contract. You need really thorough contracts. Include if the provider outsources their own work and services, and that actually frequently happens with IT suppliers or IT services. Make sure that they do understand that this is expected, what is expected regarding documentation and adherence to requirements, and re-audit. You will need to have your own business in order to make outsourced work in order. Only then are you able to state the requirements and the expectations that you have for the outsource partner, because you cannot expect them to do more than you state. Data lifecycle. Validation, including assessing risks and developing quality risk mitigation strategies for the whole cycle. Well, we covered validation a few slides ago. What I did not mention there was that validation shall be risk-based. Data itself also needs a risk strategy. This includes controls to prevent and detect risks throughout the steps of data generation and capture from an instrument or from other sources. Data transmission between systems, data systems like LIMS or production management system, between LIMS and instruments, between all other computer systems that hold GXP data. And while, for example, the financial systems don't hold GXP data, they hold tax-related data, which also will have to be under control from a legal point of view. Data processing. How do we massage our data, calculate them, report them? Data review. Here we also need SOPs to state who, when, why, and how data shall be reviewed. Then, data reporting and handling of atypical and invalid data, data retention and retrieval, and data disposal. We need to think about all of this because this is the whole data lifecycle. Data integrity risks are most likely to occur and be highest when the data processes or specific process steps are inconsistent, subjective, open to bias, unsecured, unnecessarily complex or redundant, duplicated, undefined, not well understood, if they are hybrid, based on unproven assumptions, and or not adhere to good documentation practice. A lot of data integrity risks here. Data review is ultimately the management's responsibility, but this is not very useful unless everybody adheres to the standards and do their jobs. The review and, as appropriate, evaluated statistics after the completion to determine if the outcomes are consistent, if they're compliant with established standards. They include all data, also atypical, suspect, rejected, reported data on electronic data, include the original records. The main difference from 21 CFR Part 11 work we did around 2000 was that 21 CFR Part 11 was a job to be done. Data integrity is, unfortunately, an ongoing, never-ending task. It's a lifestyle more than a job. Now, what is CAPA? This is another acronym meaning corrective actions and preventive actions. CAPA is included in the GXPs as well as in ISO 9001 and 17025, to mention a few. What it means is that we shall not only correct mistakes, but also assess if anything can be done to prevent them from happening the next time. For example, the instrument failed to give the correct results, and I'm not talking about that the results were out of spec or something, but about other problems like the blind did not meet the expected result. Root cause was found to be that no system suitability test was run or something was not calibrated. How do we make sure that this does happen another time? Or said differently, what preventive actions can be made? We include the missing part in an SOP or inside the method or whatever is appropriate. Needless to say, document the whole error and actions, CAPA. For issues found, for example, errors, you need to assess the impact on patient safety, product quality, information reliability. You need to interview people to understand the nature of the failure, and people includes technicians, supervisors, QA, management staff. Need to find out what could be done differently to prevent and detect the issue sooner, which also is the underlying root causes. I hope you learned something during this presentation. At least you now have a grasp of what data integrity is all about and which standards apply. Even if your organization do not have to follow that standard, has a lot of useful information to help you understand the GXPs and vice versa. The GAMP guide on data integrity is also very useful and should be read and adhered to. Granted, the text there is not a requirement, but the FDA strongly advises that GAMP 4 or 5 should be used, and I'm sure that would also be true for the data integrity guide. And for those of you who have a LIMS or plan to purchase one, the LIMS section here in this presentation will hopefully be a good source of input to your implementation or changes to the LIMS. Participants in this meeting are welcome to contact me for short questions, and short answers will be free of charge. But as I work as a consultant, everything requiring a very long answer or assessment will be a job for me and will be charged. Just ask, I'll let you know if it's a long answer or not. This was really my presentation, but I have a few little extra information for you in case you're interested. There are guidance for good data and records management. There are some references. There is an excellent book on data integrity and data governance by Bob McDowell. So if you're interested in data integrity and data governance, this is really the book to buy. US and EU pharma regulations are information here. GAMP is not gamp.org, but it's ispe.org, and has a lot of good guides, both the GAMP 5, the ISPE GAMP Records and Data Integrity Guide from 2017. There are several other guidelines to GAMPs regarding special fields like testing, validation of lab instruments, etc. There are some standards organizations here in case you want to purchase or look at those standards. ISO, ASTM, IEEE. I have some examples for juicy warning letters in case you don't really believe what I've said is necessary. One from Fresenius Kabi in Germany in 2013. These warning letters are from the FDA, by the way. Samples, data, and results outside of the local systems. They have unofficially pre-tested samples with selected reporting called test, trial, and demo. Out of specifications or undesirable results were ignored and not investigated. They retested without reason and only passing results considered valid for release. They had some unauthorized changes of electronic data permitted. Management were unaware of impact on API. The risk management was not functioning. They were combining an out-of-spec batch with another one to make it pass for impurities. Combinations of inadequate SOPs, inefficient training, and corporate audits failed to identify identifications. I would not like to be the receiver of this one. There's another one from India, 2014. The documentation was written after it was done. They backdated weight printouts produced after analysis where it was used for calculations. They failed to detect and investigate the inaccurate data found by the inspectors. They have unauthorized access to computer systems that could make changes in master production documentation possible. The users were able to delete data. The audit trail is disabled for several systems. Shared logins for several systems and operating systems. There were no backup of standalone GCs and no control that backup has been complete. Another one from India. Incomplete lab data, missing raw data. Trial HPLC runs stored on separate drives on personal computers, different from the normal runs. Original data files were deleted. QA unit relied on incomplete records to make batch decisions. Unauthorized personnel with access to records. Analyst logged in with admin account and no password, and the audit trails were turned on and off. Auto-titrator without named user accounts, no audit trail. Destroy original batch records and backdating revised replacement pages. Not very much like GMP. One from Italy, 2017. The lab records may be incomplete. Original and unofficial and uncontrolled spreadsheets on shared computer drive. Later transcribed to an official form. The original data showed failing results, but reports show passing results. Photocopied blank out of specification form and rewritten data. Critical lab results were shredded. Another one from India, April 2017. The HPLC software for impurity permits inhibit integration without scientific justification. Audit trail functionality was disabled. There was no review. It was exported to Excel and manipulated. There were no true copies or original records. There was uncontrolled systems used for out of specifications. missing scientific controls to prevent data deletion or alteration, requested records found shredded in the trash. These are just a couple or a few of the warning letters that people have received. There are plenty more, but I think these will prove that the FDA is really looking at data integrity these days. Now we can do the Q&A portion.
Thank you, Siri, for your informative presentation. We are now going to start the live Q&A portion of the webinar. So if you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left-hand side of your screen, and we will answer as many of your questions as we have time for. So let's get started. We've already got some questions coming in from the audience today. So Siri, we'll go ahead and start with this one. One audience member goes, "We don't have a LIMS and we don't plan on getting one. So what do we do then?"
Well, if you are working on a paper-based lab, what you need is a lot of SOPs, and you'll need your logbooks for all the instruments and for how to create your reagents, etc. Basically what I've written for the LIMS, but you need to have it on paper instead. So it's not really a big difference in what you need to document, but there is a big difference in how you need to document it and how easy it is to document.
Great, thank you so much. Moving on to our next question. We already have a LIMS. How can we check if this has been implemented well enough?
What you will need to do is to go through with my presentation in your back head and see if you have actually covered everything that is needed, like all the metadata for the samples, for the analytical methods, and all of that. So you need to go through that again and check that. I think there were quite a few ideas for you there, and even if I only covered a couple of different analytical methods, for example, I'm quite sure you understand what I'm looking at, and you can do that with your own data.
Thank you so much, Siri. And it looks like we have time for one more question, so we'll go ahead and go with this one. An audience member asks, "We plan to get a LIMS. Which one is the best, and or how do we choose one?"
Well, there are plenty of LIMS systems on the market, and there is not one that is the best. And I think the reason there are so many on the market is that they're all the best for somebody. And how you choose which one is to write your user requirement specification, and you will send that out to the vendors, LIMS vendors, and you will get the answers back. And the one that seems to fit your needs best is the best one for you. And your neighbor may have another one that's best for him or her.
Thank you, Siri. Before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information that you provided at the time of registration. We would like to once again thank Siri for her time today and her important research. We would also like to thank Laboratories and our sponsor, Sartorius, for underwriting today's educational webcast. You can view the webinar on demand. Laboratories will alert you via email when it's available for replay. That's all for now. Until next time. Bye-bye, everyone.